Hello.
Hello.
Hi, how are you?
Hello.
Hi.
How are you?
Well,
I had a blank screen. None but me.
yeah.
This time you guys slept in.
I think I went in the wrong room for a minute, and then I came back out and realized.
Oh,
Yeah.
okay. Okay.
So yeah. So again.
See, Alon doesn't have much laundry.
Oh yeah.
I've joined at 6 a.m. before, and realized that you're Central Time.
Yeah.
Hello, Susan.
Hello, Susan.
Hi.
Oh,
All right. So yeah, welcome to the meeting, Alon and Dick and Susan and Wataru and Morgan. So yeah, today we'll talk about maybe some GsoC updates. There's Harikrishna. So and then we'll get into some other things. I know we've had an email conversation about diatoms and working on diatoms. So maybe we'll go over that a little bit, and then some updates, and then we'll maybe do some papers. So, did we have any updates from anyone? Did anyone want to?
No. I can tell you one thing. We're thinking about aberrant diatoms.
Okay.
Which are typically, they're often used as indicators of polluted water.
Okay.
Stuff like that. And I just recalled I have seen electron micrographs of basilaria that are aberrant. Okay?
Yeah.
And I don't
aberrant?
Yeah, they're not straight. But I don't know that anybody's ever done movies of them.
Right.
Okay. Alon, maybe you can do a search for us.
Oh yeah.
See if you can find a movie of aberrant basilaria. That would be curious if they apply differently from straight back and forth.
Yeah.
I did go to some movies. Apparently, there's a YouTube channel about like microscopic like life or something. I don't know if you know this project. And they kind of like video like microscopy.
Journey to the Microcosmos.
Yes. Really good for me as a person of kind of like have like no knowledge, and they kind of like
That's okay.
Yeah.
No presumptions.
I love that channel. They really go into how they make their videos with the microscopy too.
Yeah. Yeah, I can also give like an update on the conversation and what like my kind of progress.
Okay. Have you resolved what we've been saying?
Yeah. So so kind of like went into a conversation with Dick and Thomas. Thomas is I didn't know him, but he's a researcher from Germany. Something.
He's actually what do you call it? An aeronautical engineer who I think is retired.
Oh. Okay.
Okay.
So he's retired, so he has time to
He's
research.
a retired engineer who's his math is over my head. So.
Yeah. So he's helping to give me up to date about like diatoms and like maybe like going into this research like with us.
He's also trying to twist your arm to get you to grow something.
Which I don't know how. Like the guy like
ask him how.
Or something.
Ask him how. It's really not hard. I've done it.
Oh yeah. I'm kind of guessing it's probably gonna have like a glass to kind of go in the estuary nearby, like in the river, and they kind of collect some samples and somehow I don't know.
I was told to use soil and then just wet it down, make a slurry, and then make water slurry, and that will grow diatoms. The soil will feed the diatoms.
Unless it's full of pesticides.
Well, you have to get some. You have to be careful.
Anyway, they're not hard to grow.
And the good thing, he sent some high resolution. high resolution like video clips of diatoms within like with kind of like some of the requirements that I wanted for tracking, which was kind of like to get the camera focused on one spot and have like maybe like a high kind of like a bit of like high wide aperture, and also like the resolution just to be like one focus and the resolution like to be better. And that's good like for start, and to kind of like for me to kind of like maybe gauge like what the possibilities that I can work with the tools that there are like already in the digital Basileria repo.
Yeah.
So I downloaded the repo. I kind of like installed it locally, and I installed all the kind of like prerequisites. It kind of took a bit of time, and then I trained the Detectron segmentation and object detection neural network. And that gave some, I think, not like results that kind of like I'm not like happy with, because for like tracking, if it's smooth, I'm kind of like wanting something to kind of like get all of it kind of like maybe in a high precision, which is a bit hard in instance segmentation to kind of differentiate between the different instances of the diatoms. And yeah, we're kind of like, I think after that, we chatted a bit in the email, and we said that we're going to do a meeting.
You asked about features. When we did single diatoms, which proved to be jerky, we used the outline, and we went through, if you look at the paper, I think we went through about 8 different algorithms for estimating the centroid of the diatom based on its outline. And we were able to get sub-pixel resolution for doing that. Okay, because as long as the diatom doesn't turn or roll over in some sense, then you have the same view, and therefore you can track it easily.
Yeah,
relatively easily. But so we did an analysis. Now we also took some plastic beads and sintered them to a glass surface, I think. So we would get an image of something that was not moving. Okay? And despite that, it moved.
The diatoms? Or the,
no, the beads.
The beads.
Yeah, and that gave us a baseline for errors in the system.
Yeah.
Okay, because
in the tracking system.
In the tracking, we were tracking so low that we were probably full-time limited. You know, I don't know. You can try this experiment yourself. Dark adapt your eyes, and try to look at a small object in a dark room, and you will see that it moves. Okay.
Because of I was guessing because it's like the just a measurement noise in the measurement that we are getting.
Yeah, but no, the noise in the photon fluctuations can lead to apparent motion.
Okay.
Okay. But you can actually see that effect if you try that experiment. Because our eyes are fantastic. We have, I think, around a billion-fold range of brightnesses, especially if you get dark adapted. Okay.
So I need to kind of like read this like paper. I don't know if you have this to kind of like to send it to me, or if it's in the repo.
Yeah, it should. Did I give you access to my papers?
I think you did. Yes,
yeah,
I think
it's in there. The first author is Sabunku. I'll write his name.
I'll write his name. Yes. Okay. Thank you. And because what I was thinking later on to kind of like maybe do like a corner detection, I was kind of like asking a question there because they have all these kind of like edges on the diatom.
Okay, it's in the chat.
And if I can track like each corner on the diatom and it's rigid, that's going to help me like to derive a center of mass from it.
You asked about the dots. The dots are probably pores which are focusing light a bit. They are.
Because it's a glassy exterior. So you're saying like there's like maybe some.
Yeah, it's glass. And we've looked at diatoms. They have very fine structure. The light microscope does not do it justice. If you want to see the fine structure, you need a scanning electron microscope. You want to see the details, but some of those details will show up in light, and as long as they're constant, which you can judge by eye, there's no reason not to use them for tracking if you want.
Yes.
If you want.
Yes. And because they're kind of like also staying like relatively, I think, like stable to my eye, like within the diatom, so that's kind of like a good feature to kind of like if it would be moving like within the diatom, that's gonna be like less.
Yeah,
effective.
no, I don't. Well, some things will move, like the chloroplast possibly. But here, look up scanning electron microscope of diatoms, and you'll see some of the fine structure.
Okay.
SEM.
SEM.
So the light microscope, it can't see. It doesn't have the resolution of the scanning electron microscope.
Okay.
But the diatom structure does.
Okay.
In fact, I once did a calculation that if you consider the spatial resolution of a 3D printer and tried to print a diatom so that you could see all the structure that's visible, the diatom would have to be printed 50 meters in diameter.
Oh, okay.
Okay.
Yeah, because of the resolution, because the
resolution of the 3D printer.
Yeah.
Okay.
Yeah, that's kind of like it's really.
It shows you how much detail is available in a diatom.
Yeah. Yeah, and what else did we? I think Thomas also like he wanted to go to like a synthetic video to start with that to see like what noise are we getting and gauge this noise first, and then that's gonna help us.
That might be a good idea. Yes. Synthetic video, which you make smooth, then you can see if you can detect it smooth.
Exactly, because like I think he did something, and then he got like some noise in the velocity of the y and the y-axis or something. But yeah, just to see like what, and I'm kind of like I don't know like if I think we're gonna have to decide of a definition of kind of like what smooth is, or like if it's kind of smooth to kind of like what we got like for now, or like we can say like that's the smoothest that we can. So or like
okay, look, we have that definition. It's in the two papers by Sabunku and
okay, good.
Paper by Edgar. Paper by Edgar is 1979. She did it she took movies at, she analyzed at 10 frames a second. We made movies at 890 frames a second. And we got the same result that she did.
Okay.
But for single diatoms, the motion is very jerky.
Yes.
Okay.
Okay, cool. Yeah, so I can get that background.
Yeah. Do you have the Edgar reference?
No. I thought is that not also like in the papers if it's in your?
I said it should be mentioned. Yes, it should be cited.
It's a bundle.
That's a 1979 paper.
Okay.
Okay.
Yes. And yeah, I think that's where we got so far.
Okay. I'm trying to think if you simulated I think we might have done that. If you simulate a small spherical object or that is not moving, you can get it to move by simulating photon noise. In other words, by having some statistics in the picture. And I'm not sure what a realistic simulation would be there, but then it would move, and that would give you a baseline of something that has smooth motion because it's not moving at all, but nevertheless seems to move.
Yeah, it depends on where you simulate it, like how and also like you would simulate like this photon kind of like movement.
Well, no, it's not photon movement. It's the number of photons arriving in the image.
I thought it's more like a perception thing, that we perceive it to be moving, but it's not.
No,
No.
not necessarily. That's why I said do the experiment. You see, if you're in a dark room with very little light and you're like dark adapted, then your
Yeah.
eyes are picking up. You can actually see single photons. You know, if they're directed at
But
you.
how would they differentiate it if it's my perception or
By simulation.
my simulation? Oh, yes. Well, I'm gonna have to simulate both of them. So that's
Okay, no, I'm just saying that shows you a property of your eyes.
yeah.
Okay, but if you have a noisy image of something, and you have many noisy images, and then you try to get the centroid of the object, the centroid will move, even if the image is not, if the original object is not moving at all.
Yeah. Yeah.
Okay, and that gives you a baseline for what statistics to expect.
Exactly. Yeah.
Trying to discriminate smooth motion, because that's smooth motion. I mean, you know
moving
it's not moving.
Yeah.
Okay.
And we want to know like a smooth movement, not just like not moving.
Yeah, but zero movement is movement
Yes.
of an object that's not moving is a baseline.
Yes.
Okay.
Okay. Yeah. So so that's kind of like where we are.
Okay.
Yeah.
Sounds good.
Yeah, it sounds great. Thank you for that update. I had Sushmanth put something in the chat here. Actually, Rutherford did this thing in gold foil experiment. He was in darkroom for more than 1 week to do gold foil. So this is the gold foil. Actually, Sushmanth, if you want to introduce yourself and tell us more about that, could you do that? I don't know if you can speak. Yeah, there we go. Yes, you're audible.
Can you hear me?
Yes.
I'm from Amrita Vishwakarma. I'm junior of my dev. He was my senior. He introduced me to DevoWorm organization and all. I found it so fascinating. Actually, when Dick was talking about being in a dark room, Alan was telling like that thing only Rutherford did in gold foil experiment to see those photons, like they are moving or not. That's the reason I have kept that message.
Can you describe the experiment?
Photon experiment actually. When with that gold foil, like he was discovering nucleus, he was sending photons through that gold foil. They will reflect back when it was hit by a nucleus onto the another coil. To see with this thing with exact eyes, he was being in a dark room for more than 1 week, and he did that experiment to know that thing.
You could actually see the foil move from a single photon.
No, not by single photon. He used to eject around 20,000 photons like that, and he used to see like only. This thing was taught to us in 12th standard for an experiment, gold foil experiment. So I thought it was related, and I kept that message.
Okay. Can you get a reference to the original paper?
Yeah, I'll share.
Okay. I mean, I wasn't familiar with this one. Okay.
Yeah. Well, yeah. Thank you, Sushmanth. I think you've been involved in some of the DevoWorm, one of the DevoWorm pull requests and issues. So
yeah. I started working on it, but I thought like I have done incomplete work, so I closed that pull request. I was able to start working on that issue, which was issued by my. I was learning the architecture of full complete DevoWorm, and I need to meet my today for explaining about all stuff how it works, but I couldn't go. He was still, I think so. Tomorrow maybe I will meet and I will start working on that issue.
Yeah, that would be great. Well, thanks. Yeah, welcome to the meeting.
And by the way, Alan, sorry, yesterday I was okay.
What was
sorry, that? By the way, yesterday I was busy with another think that's the reason I couldn't attend the meet.
Oh, no problem. Yeah.
Yeah. Yeah, we thought of kind of like meeting up to kind of like to fix that issue because Sushmanth kind of like he did something, he kind of fixed it, and then we could just like maybe just check it out and then upload it. But so anyway, if you're finishing it today, then no worries.
Okay.
Okay, yeah. Well, thanks for introducing yourself. Yeah, and if you're interested in something further, you can contact me, or you can put up, you can put a comment on the issue, or you can open your own issue in DevoWorm, and we'll respond to it.
Absolutely.
Okay, I've got a question for Alon. Did you have a look at the possibility of trying to enhance the jelly stuff around the basilaria?
No.
Okay.
No.
We don't know if the elastic properties of that, or the viscoelastic properties of it, might influence the cells and therefore provide a feedback mechanism between the cells. Because right now, there is no obvious way that the cells can communicate with one another, but nevertheless, their motion is nearly synchronous.
Yeah. I understood that it's a problem, that we don't know how they communicate, but it's synchronous.
There's speculations that they do so by light piping, could be mechanical through this jelly that's on the outside, we don't know. Okay, so an attempt to visualize, I don't know if you can see it. I can just see some particles moving, suggesting that they're part of a jelly mass. But enhancing it maybe with histogram equalization or something like that might show it up.
Okay.
Yeah.
Yeah, the parts that are moving like on the surface of the diatoms, or like
Bradley, there was also an interesting movie that came across. I forgot which one, but the synchronization is only partial. And there's actually an image where two parts of the same colony collide into each other and stop, suggesting that there is a means of some sort for long-range communication between the cells, not just nearest neighbors.
Okay, yeah.
Okay, and that might be a hint that the behavior is different from a simple nearest neighborhood model, which is probably what most people are thinking about.
Right.
Yeah, that's what I was also like thinking, with kind of like that they talk like just between the nearest one and then kind of communicate.
Yeah, but there might be some long range communication.
Yeah.
Right. Well, that's interesting. Yeah. Is this in the videos that Thomas made, or is that a different set?
No, I think it was one of the online ones.
Okay.
Can you dig up which one I sent you?
Who did you send it to?
Where you sent me? I'm not sure who sent it to who. But
did you send it to me, or like?
Yeah, it was a movie which showed two groups in the same colony colliding with each other and stopping.
Okay.
They actually stopped. And
I don't remember, like I only remember like Thomas sent me some videos, so maybe.
It might have. So I don't know. The overall shape is zigzaggy, and the zigzags. This was a remarkable movie in that the zigzags actually collide with each other.
Yeah.
Okay.
Nice.
So it's just a hint of what might be happening. I don't know.
Yeah.
If you look at the stuff that Thomas has written and Bradley on the motion, they're getting very sophisticated.
Yeah.
On the behavior of these things and the intelligence of them.
Yeah. Yeah.
Cool. Okay.
Okay.
All right.
Oh, and if you want to hunt for diatoms for basilaria tends to float in the water. If you want to hunt for it, you'll need what's called a plankton net. It's a very fine meshed net. Okay, and it's built so it concentrates water into a small bottle, and that way you can sweep a lot of water and collect the diatoms. And if you're lucky, you'll get Basilaria. Okay. I had one student who spent a whole summer hunting all around the province for Basilaria, and then she found it right nearby.
Nice.
Yeah.
Got to rule that out. Okay.
Okay.
Yeah.
And Thomas has similar problems. Sometimes he gets lots of Basilaria, and other times he can't find it.
Yeah. 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 Thanks for that update and everything. Now I wanted to get to Wataru and Harikrishna for their updates on their projects.
Okay.
If we have, who wants to go first?
Hello.
Hi, how are you?
Yeah, so I'll show what I worked on this week.
All right.
Let me share my screen. Yeah, so this was the model which I created, ellipsoid which I created using the Python code. So the next step was to map the image. So I wasn't able to do using the code itself because the results were not nice. So what I did was I loaded the model into Blender, and then this was the best UV unwrap which I could get, which perfectly fits over the image, almost perfectly. So now what I'm doing is I UV unwrap in Blender and save the model, and now I'll be using the saved model in my application. So there'll be options in the application to change the textures, which is the the image of the embryo. So if you arrange it like this, the image will be mapped correctly. So this is what I worked on, and I also worked on parts of my application, which is the final result. So now currently what I'm doing is finishing up the application and also writing the README file for the GitHub repository.
Okay, sounds good.
I also have to write all the instructions to install it for any other user to use it.
Okay. So when you have it, so you're building this application that people can use, do they have to have a data set ready to put into the thing? Or, I mean, I guess we can provide them with data, but how are you? This is a 3D model of the embryo. So you can give the image as an input, which includes two opposite sides of the embryo. So it will map on that 3D object accordingly, and we can change the image according to the stages of the embryo. So that's what the final application will be. All right, and then the images are kind of like prefabricated, or like if you wanted to put in some new images later, is there a way to do you have to reference them in any way, or do you just put in? How does that usually work?
There will be option to specify the image location. Using that, we can put new images too.
Okay, good. And the image or Oh, go ahead.
Only thing is that it has to be in a specific. The image has to be in a specific ways, which I'll write in the instructions. That's it.
Okay. And so then the application itself how is that being written?
Right now I'm writing it in Electron.js using JavaScript. This is because I also want to deploy it in web too, so I can use the same code for web and desktop application.
All right. Yeah, that's good. And so you think you're on track for Now, the submission deadline, I think, is September 7th. And so are you on track for that?
Yeah, I think I'm on track with it. I'll be able to complete it entirely by September 7th.
Okay, that's good.
And I also want to, there are some limitations though, which I'll be working maybe later after the submission.
Okay.
Harikrishna, could you please put your picture up again?
Yeah.
I've got a question about. I guess the question starts with, is there a mathematician in the audience? Okay. Okay, now if you look at the picture on the left side, it's a flat picture which is a disk. And what it reminds me of is a flat drum. Okay, if you're familiar with drums, the modes of oscillation of a drum can be expressed as Bessel functions. And the Bessel functions are over a circular plane. Now, on a sphere, we're dealing with spherical harmonics. So my question is, given that these are projection images of a spherical surface, is there some relationship between Bessel functions in the plane and spherical functions, spherical harmonics on the sphere, which we could exploit in order to project the image? Okay. Now it's well beyond my math.
I'm also not sure about the math behind.
Okay, but you see, there's a possible relationship between the Bessel functions and the spherical harmonics. In some sense, the Bessel functions are projections of the spherical harmonics.
I am not familiar with both of those terms, so I will have to read about them first.
Morgan, you're familiar with spherical harmonics and Bessel functions?
No, neither of them.
Neither of them. Okay. Okay. Yeah, it's beyond my college math.
I can look it up as well. There might be someone I could ask about this.
Okay, you see, there's possibly a relationship between the two kinds of functions that would be exploitable.
I'll read about it, and if I find anything, I'll let you know through email.
Okay.
Yeah.
Wait, for EEG, I was also taught by an aeronautical engineer for this for spherical harmonics.
Okay. Were you taught by a drummer about Bessel functions?
No, but they have come up. Drumming has come up. Yes.
Okay, so
but Paul Nunez wrote Electric Fields of the Brain, and which is kind of considered the
seminal
text of final questions, and his background is actually aeronautical engineering, which is kind of
okay. Yeah, maybe we should pull Thomas in, because he's got that background.
Well, that's good, Harikrishna. Thank you for the update. So, yeah, please keep in touch as we're coming towards the end of the project, and I'll have to contact Karan as well and make sure that you're coordinated for the final submission and everything. Well, I'll talk about that more, in the Slack, and we'll sort of coordinate that, make sure that everything is okay. Yeah, it's good. Okay, and there's a link by Morgan. I think that's that book he just mentioned. Yeah. So Wataru, hello. How are you?
We are refactoring the serial tracking paper, and I'm working on the inference and evaluation part, and I'm starting with separating the datasets from the mainstream, and I'm separating into the objects. I will soon make a PR in the inference part.
Okay.
But the problem is that the computing process takes too much time. So we might not have time to test our refactored cores.
Okay. Do you think they'll work though? I mean, I don't. Yeah. Well, I mean, so you guys are pretty much on track, except you don't know how much you can test things.
Yeah.
Okay. Well, I mean, you can do like, I guess it just has to run, and it doesn't need to be like perfect. So I wouldn't worry too much about testing. Just make sure that it can run. At least just get it to and then that's fine for now. And then after the submission deadline, then you can work on it more if you'd like. That's always the way these things go. They give you a certain amount of time to get to a certain point, and then that's it. But we have the deadline coming up on the 7th once again. So do you think that you'll be ready to submit by then? And both of Jiahong and yourself have to submit separate, reports. You have to submit something, and you have to have the documentation and everything. So do you think you're on track for that?
Yeah, I think I can submit something.
Yeah.
But maybe. I think. I'm sure I can submit something to Google.
Yeah, that's good. So I'll be in touch again over the course of this next week. So it's not that long. It's maybe a little bit more than a week left. So, work on the documentation, decide what's in the thing that you're submitting, and then, we'll go ahead and I'll review it maybe before if you want, and then you can submit it, and then, you can put what you and Jiahong have been doing into a larger package, or I guess it's all one package now. It's just a matter of what part you did. So like you focused on stage 1 and Jiahong focused on stages 2 and 3. I guess you kind of worked on together, but you can just stress what you did on your own, and then put that together, and then we can figure out, like, after the 7th, how we want to put this whole project into DevoLearn, because we're going to eventually incorporate this whole thing into the DevoLearn platform. So that's good.
Yeah.
Well, yeah, thank you for the update. That's good.
Yeah, thank you.
All right, so, and then I wanted to mention one more thing about that, about the graph neural network stuff. So I've forgotten about this a little bit over the summer, but I think we can still do it. So there's this conference that Jiahong brought to my attention, and this conference on learning on graphs. So it's this conference that I don't know if it's being run as a parallel to. I don't think it's a parallel to anything, actually. I think it's just the conference. I think it may be concurrent with NeurIPS. I'm not really sure. But this is what we want to target with this work. So when Wataru and Jiahong are writing up their stuff, we can take things from that write-up and put it into this paper. And then maybe put together why we want to do this. And so we would have a nice paper, I think. and it'll be kind of already written up. So the thing is that there's a fairly quick turnaround here for this. So, when you finish on the 7th and you finish your write-ups and submit things, on the 9th, there's a submission deadline for an abstract. So that's no problem. That can be done I can work on that in advance. And then the submission deadline for the paper, I think, is the 16th of September. So you can see abstract submission, 11 days. And the way they do this in these conferences is they do an abstract, and then I think a week later they do a paper submission. So it's kind of an odd system, but basically they want to make sure that you're submitting an abstract that isn't off the wall, something they obviously wouldn't want, and then you can submit your paper. And then, the conference is on December 9th. So this is we'll get a final decision by sometime in November. Actually, I think a submission decision in October, and then some revisions in November. And then there's a final decision. And then the conference is December 9th. So this would be nice to have as a, something that just is an immediate output of this summer project. So these accepted proceedings papers, I don't know what the acceptance rate is, will be published in Proceedings for Machine Learning Research. So that's nice. Full proceedings papers can have up to 9 pages with unlimited for reference as an appendix. So again, all the work that Ji-Hung and Wataru have been doing, that's probably going to be quite a bit. It'll probably not be too hard to get near 9 pages on that, along with some information about why we're doing it. So I think the registration is free, and it's on a virtual conference, so it's very little overhead on our part. Just sign up, I guess. And then there's an overleaf template which we can use as well. But those are the details we'd work out later. So yeah, this is, and then I looked at the subject areas, and it looks like we kind of fit in here somewhere. The only caveat I mentioned is that maybe they're not, maybe it's a little too biologically oriented, but I don't think that's going to be a problem, because really we're talking about applying this to biology and not the biology itself. But we'll see. So that's I think, yeah. Go ahead.
Could you put the URL in the chat?
Yeah. Let me get it. So that's the log conference. Unless this is the call for papers. There we go. All right, there you go. And that's I think an immediate goal for this. And then longer term, longer term being soon after, we'll try to integrate this into the DevoWorm platform and see how that goes. And then there's also a DevoWorm sort of technical paper we'll probably update from there too, because if we update the software, we'll update that. So yeah, that's the conference call for papers. So that's about that. Now I know that Harikrishna had to leave. Thank you, Harikrishna, and thank you, Sushmanth, for attending. I also had some things on diatoms I pulled up because I knew we'd be talking about them. This might be a benefit to Alon, because I don't know how much he knows or he's seen of diatom phenotypes and things like that. So there's a Wikipedia page on diatoms. This is their, it's a large group of algae. It's very diverse. They have the silica phenotype, the silica walls of the cells. So they can be single-celled or they can be multiple-celled. So this is the morphology. They actually have these different types of morphology. The pennate one, which is long, and the centric one. So the bacillary are more pennate. And then this is the structure of a frustal, which is the where they have these micropores, and they have all this detail that Dick was talking about when he said that the detail is so fine you need you would have to, if you were to do 3D printing job of like the anatomical detail would be like 50 meters in size, because all these little different Go ahead.
Those pores have pores.
Oh, the pores have pores. Yeah.
Right.
Yeah. So these cells are like less than 100 microns, right, Dick?
Yeah, the smallest I've read about are 2 microns.
Oh wow. Okay. So yeah, and this is a shape classification. So they're the rod-shaped ones like this. This is kind of like basilaria, but they have the pores along the side and some other features. Then they have the flake shapes, which are like this. So you can see they're really diverse in terms of their shape. And then the 3-dimensional shapes. So some of them have like depth. So when we're looking at the basilaria videos, we're looking at the 2-dimensional plane, and that's enough to characterize the basilaria. But these are 3-dimensional ones where you have depth that's also important. So that's let's see. And so then their behavior, which we were talking about, and this is just the Wikipedia page. So this isn't like complete by any means, but they talk about how and I know Dick has written some work on like the different modes of movement. So one of the things about diatoms is that there are a lot of theories about how they move, and there are a lot of potential mechanisms, but no one actually knows, and it varies by species, I imagine, but no one actually knows what the definitive mechanisms are. Like that's why we're kind of like why we're doing this, because there's we have the different modes of movement, and we don't know. I mean, we're not going to get into like the mechanism through just observing videos, but just characterizing the movements are enough, and then knowing exactly what the mechanism is, that's actually quite hard. So yeah. And so there are a lot of images, like people have drawn these for a couple centuries now. I think like the first diatoms were sort of identified in like the 18th century, and people have drawn these out. So before they had images, cameras, they had their hands and they just drew things. So this is this sort of drawing under, looking under a microscope and drawing out the detail. And then, people have done this in various ways, just observing these different cell types, and that can give you some information too as to what's going on. They have a interesting worldwide distribution. Their ecology is very diverse. Also, their life cycle is really kind of wild. And then there's this article, Scientific American article, on. Let me put this in the chat.
Yes, thank you.
Yeah, so this is a Scientific American article on diatom, or the trouble with life in glass houses. And so a lot of this is, this kind of goes over this diversity of diatoms. They're made of the silicates, so they're kind of glass, they have these glass houses. And this is a nice article on some of the, what's going on with this. It's at a Scientific American level, so it's not like really technical, but it kind of gets into some of the details of what makes these organisms special. So that's good. We have some papers. If you go to the DevoWorm website, there's some papers on diatoms that we've done. I think we've done 2 at this point. One on kind of like the behavioral mechanisms, and what Dick was talking about with the intelligence part, and then one on like do you think you've already read that, on the cell tracking and some of these other things that the ways to sort of characterize the phenotype.
Nice.
Yeah.
And I am going to follow up with Exploratorium on their materials and their like resources, because again they are actively studying diatoms.
Okay. Yeah.
Okay.
Really?
Yeah.
Oh, nice.
So I've got some pictures from my phone that I can get out, as well as like their pictures of their microscopy setup. But like because they are a community resource, or trying to be an educational resource.
Yeah.
I'm gonna make a time to set a time to talk with their researchers, and maybe get them to come talk.
Oh, that'd be good.
Yeah. I'm also going to reach out to the one C. elegans lab that I know, and Lausanne, and I'd be really curious to see if they could use some of the software.
Oh yeah. What C. elegans lab is that?
That'd be in Lausanne. So that EPFL.
Okay. Oh yeah.
Yeah.
All right. Yeah, that's great. Yeah. So, did we have anything else before we move on? I think I want to get into papers next, but if we had something, okay, Dick had some references here.
From Basileria.
Yeah. So Dick also posted a link to his paper on the whimsical history of proposed motors for diatom motility. So this is the paper I was talking about with the different mechanisms for movement. And yeah.
Basileria was one of the first diatoms discovered in the 1700s.
Yeah.
And this last reference includes a translation from ancient Danish to English.
Oh.
Yeah. Okay. Yeah. So let me share my screen again. And I'm going to go through some papers now. We have... here we go. So in the past, I think it's been several meetings now since we got into this. We had this thing about push and pull morphogenesis. So I think it's in the recordings, if you don't remember it, but it is this series of papers where they talk about this mechanism for morphogenesis, where there's like bending of the tissue, and then there's a feedback that triggers gene expression, which triggers more bending, and then the definition of different things. So there's this there's this model system in the chick embryo where they have this stippling pattern, and this is the type of morphogenesis that you see. It's very different from the reaction to fusion morphogenesis of Turing, which is chemical morphogenesis, where you have morphogens, and they set up these gradients. This is different. This is where you have these spots where there's buckling, and then the buckling is reinforced through different mechanisms, and then you get this stippling pattern. So it's very different. Then there, I think we did a couple papers on that, and then now there's this other paper I wanted to talk about within this sort of the in the scope of this. And this paper is from the International Society of Biofabrication, or I guess it's Biofabrication Journal. This is an accepted manuscript, and this is calcium supplementation of bioinks reduces shear stress-induced cell damage during bioprinting. So a little bit different take on it from some of the model systems. But this is an interesting paper, kind of with more of a bioprinting aspect to it. So the abstract reads, during bioprinting, cells are suspended in a viscous bioink and extruded under pressure through small diameter printing needles. The combination of high pressure and small needle diameters expose cells to considerable shear stress, which can lead to cell damage and death. Approaches to monitor and control shear stress-induced cell damage are currently not well established. To visualize the effects of printing-induced shear stress on plasma membrane integrity, we add this to FM143 to the bioink, a styrol dye that becomes fluorescent when bound to lipid membranes. so does the cellular plasma membranes. Upon plasma membrane disruption, the dye enters the cell and also stains intracellular membranes. Extrusion of alginate suspended cells through a 200 micron printing needle led to the increase in this FM143 incorporation at high pressure, demonstrating that typical shear stresses during bioprinting and transiently damage the plasma membrane. So this goes on to talk about, let's see. We recommend that bioink should be routinely supplemented with physiological concentration of calcium ions to reduce shear stress-induced cell damage and death during extrusion bioprinting. So this is kind of like a nice paper on talking about some of the techniques they use for bioprinting and some of these physical aspects to it. So they talk about fluid shear stress. They talk about how to make the cells more robust to this. So basically what's happening is they want to print cells on a substrate and they force it through a needle or some sort of deposition device. And one of the problems they have is that the cells break apart and then it's no longer any good. So what they want to do here is they want to utilize different different types of things to mitigate these cells blowing up when they shoot them out. So they're putting them in high pressure situations. So they're sending them down this printing needle and they're depositing them. And you have this sort of, they're doing these things with a lot of the chemicals and different things that they're doing on this. So I don't know if we have any good pictures of this process. But it has a lot to do with microfluidics and some of these other things. So this is an example of the cells here. They're staining for this thing that they're putting into the cells. They're showing the incorporation of this dye. This kind of visualizes the membrane damage. So there's a lot of membrane damage to the cells. Fluorescence with cell elongation and shear stress. So this FM143, you can see with different shear stresses that it's intense. So they can visualize some of these stresses in the cells. And then
has anybody tried looking at centrifuging and using that dye to see what happens to cells?
I'm not really sure. I'm not familiar with. I'm not really familiar with this technique. So, but I don't know. I guess it would make some sort of difference. I mean, but anyways, yeah. So this just kind of goes through this, and I can send this along if you're interested in reading more about it. Yeah. So the next paper I wanted to talk about is this paper here. This is pattern growth and control. This is a review paper, so this is actually something that kind of is maybe a little bit more directly related to some of the things that we were talking about with this other form of morphogenesis. But this is a review article on pattern growth and control and some of the things that are involved in pattern formation. So this is by Arthur D. Lander at UC Irvine. And the abstract reads, systems biology seeks not only to discover the machinery of life, but to understand how such machinery is used for control. And when they say control, they mean regulation that achieves or maintains a desired useful end. So it's like, when you have some sort of regulation in the cell, whether it be of forces or of gene products or of some sort of like tissue or something, there's this idea of control. There's regulation, which is making sure that it, is sort of in this range of functional range. And then there's control, which is this sort of desired useful end aspect. So it's like control is always making sure that it's at that what the output is useful, instead of just regulating it, like, cutting off some process and then it doesn't really have a useful end to it. So this is actually, and it has an engineering-centered approach. So this sort of goal-directed, engineering-centered approach also has deep historical roots in developmental biology. So this is something we see in engineering, and we can see in bioengineering, of course. But you see this metaphor being used in developmental biology in a way that's, maybe it's not appropriate in some ways, but that's the language that's used. Not surprisingly, developmental biology is currently enjoying an influx of ideas and methods from systems biology. This review highlights current efforts to elucidate design principles underlying the engineering objectives of robustness, precision, and scaling as they relate to the developmental control of growth and pattern formation. Examples from vertebrate and invertebrate development are used to illustrate general lessons, including the value of integral feedback in achieving set point control. So set point control is where it's the basic example of homeostasis, where you have a set point and you want to keep your system at that set point. So like for a thermostat, you keep the room temperature at 73 degrees Fahrenheit. The room temperature fluctuates, and then it's up to your climate control systems to bring it back down to that temperature or back up to that temperature. So that's the kind of thing that they're looking at here. So one of the things, anyways, the usefulness of self-organizing behavior. And we've talked about self-organizing behavior with respect to different swarms and flocks and other types of collective intelligence and other types of collective behaviors. So those are usually considered self-organizing. The importance of recognizing and appropriately handling noise. So noise is where you have, it could be a stochastic process. It could be just something like we talked about earlier, that if something's moving in a column of water just kind of at random, that's noise. There's also technical noise like static or something like that. But in biology, noise is actually very important, and sometimes it's driven intentionally to achieve certain things in the system. So sometimes there's this sort of intrinsic noise that's driven by stochastic. What was that?
Not Molly.
Oh. And then finally, the absence of a free lunch. So a free lunch is actually where you get basically work for free, or you get energy for free. And so that's something that if you know anything about, like in computer science, there's the no free lunch theorem, or free lunches are usually like, energy or work or information for free. And that's usually a violation of our principles like the laws of thermodynamics and things like that. So people are very suspicious when you say, when you mention something, it sounds like a free lunch, not just because they don't like to give out free lunches, but because it does violate some of the principles of our theories about, thermodynamics and information. By illuminating such principles, systems biology is helping to create a functional framework within which to make sense of the mechanistic complexity of organismal development. So this is kind of like a nice review of going through some of these ideas that are being maybe imported from systems biology, and bringing them into developmental biology. The mainstream of developmental biology is not typically, they don't think like systems biologists necessarily. Their focus isn't on some of these larger concepts of like control or of like, noise or robustness or any of that. So this is kind of a newish area in that field. Now, some, in like, for example, in development or in evolutionary biology, there are people working on things like robustness and evolvability, but even those aren't necessarily in the mainstream of that field. So sometimes you get these ideas that kind of enter the field in different ways, and you don't necessarily know their source. So as a consequence, people tend to misuse terms, or they tend to misapply the concept. So this is like a nice way to kind of ground people in where these things come from. So the frequency, one part here mentions the frequency and degree with which embryonic regulation, canalization, robustness, and precision are encountered and development raises many questions. So this is one of the things that's fascinated people about, like, say, embryogenesis, is that you can get, these precise things going on in development, and they're self-organized in the sense of like how we might see like pattern formation being self-organized. And people ask questions, and the question is it something that's, what are the mechanisms here at play? They're not things that you can necessarily probe from like the typical developmental biology experiments, or there's people done these experiments to show proof of concept things. But some of these things like robustness, it's kind of hard to measure that from a typical experiment. So, you have to kind of consider maybe what's going on underneath the hood. So are there common principles underlying all such phenomena? Are there conserved mechanisms? Can we explain how and why such processes evolved? So, all these questions revolve around these processes, and you could say, well, of course they do. You don't have to waste your time. We kind of know that they do, but you have all these different species, and I've shown you like these different species just of Diatom and all these different phenotypes. And the question is this a common principle underlying all these different phenotypes? All the different phenotypes that we see just in Diatoms are the same processes underlying some of these, changes in morphology or changes in behavior. And, that's still, I guess we assume that they are, but that may not necessarily be the case. There may be mechanisms that are unique for certain species. There may be some that are conserved or shared amongst species in other cases. So this is something to think about. So a lot of what we think about in terms of these universal, maybe we can call them universal principles that we mentioned up in the abstract, we think about this as sort of like a physical law. And there are physical laws that sort of govern some of the things in development. So like Fick's law of diffusion or the universal gas law, and those are things that will give you some of these products of emergence. So, we have a lot of things that exist. We can apply them to biological systems. But, and this is something I've asked myself, maybe these aren't really sufficient for really explaining what's going on in the biology. Maybe they're not like totally sufficient. Maybe they can actually give you a nice heuristic, but maybe we need special sort of corollaries of physical laws or biological systems. It's not really clear. So they have this nice diagram of control objectives in morphogenesis. You start from let's see. So this talks about feedback regulation and some of these control objectives. So you can see across the sort of these different these are embryos here. These are, they start to get wings, Drosophila wings. You get phenotypes, you get brains, you get muscle, striated muscle, you get a liver here, and you get like a retina. So you have these different things in that are developing either at the scale of just kind of like this embryo or these organs or organisms. And so you have this circle here where it tells you what kind of is most important in control. So, for example, if you're looking at organs like the eye or the liver or striated muscle or the brain, growth control is the most important thing here. So this is the mode of control that's most relevant to that scale or that type of development. Conversely, if you're looking at embryos and you have like different types of embryos, this is a Drosophila embryo, for example. This is pattern formation. So actually it goes from the embryo, different types of embryos. Actually, then you start to get into the Drosophila imaginal disc, which becomes a wing in this case. And then also in fishes, like in the phenotype, when you get like a juvenile phenotype from this embryo, pattern formation is important. So in the case of this fish, striping is controlled by pattern formation. There's pattern formation in the embryo, and then there's pattern formation in one of these organs. It's not growth control at this point. It's just these vein patterns that are being controlled. Then there's a self-organization, which is actually almost, it's pretty much concurrent with pattern formation. And this is also where you have a bunch of parts that need to come together and behave collectively or be organized into a pattern. So that makes sense that those two things are overlapping. Again, self-organization is a very general term, but generally we can use it in this area. Then boundary organized is just limited to the embryos here instead of these other things, because boundary organization is basically defines the boundaries of an embryo. So if you want to define the head from the midsection from the tail, you get these boundaries that are set up within the embryo, and they define tissues. They define different parts of what will become different parts of the organism. And so this is actually overlapping with both pattern formation and self-organization, because part of self-organization is where these boundaries are defined, and then within those boundaries you get self-organization of different structures. Then there's the scaling to size, which is smaller, even a smaller domain yet. And this is where things scale to a certain size, like the head will scale to the body size, and that will be maintained across development. So this is featured largely in different types of embryos here. And then scaling to pattern, which is where you get pattern formation, and then that scales up with the organism. And then, of course, regeneration, which is involved in, like, say, striping or some other set of organs, not all organs, though. And this is just as regenerative capacity. Can you regenerate the structure of the organ, or, something like the pattern if it's damaged somehow? And so this is where this comes into play. So we've talked about all these different themes in the group, and I think that's a nice summary of a lot of the things we've talked about with respect to some of these topics of regeneration and growth and pattern formation and the like. Then there's this, these are the two modes of organization and the control of pattern. This is the self-organized case where you get short and long range control. So this is well known in developmental biology, where you get activation and inhibition, depletion of different factors. So you get different signaling molecules, different concentrations of different morphogens and other types of gene products that are defined in different embryos. So you get this long range control, which is over the whole embryo, or short range control, which is locally. And there are these different circuits that people show in papers. So this is an example here in this figure, where you have this pulse of one factor over the sort of the anterior, I guess it's the anterior posterior axis of the organism, and you get these pulses, which might represent like a striping pattern of some type, and then this red function, which represents sort of what's co-expressed with it. So you get this pattern here, and so they combine in different ways to define boundaries to build these patterns or whatever. Then you get this pattern here at the bottom, which is this dotted pattern, which is like maybe an animal coat or the surface of a seashell, and it results from this type of expression pattern and ultimately this type of control. Boundary organized control is where you have all these generic morphogens, which are these molecules, but it's really a theoretical construct describing these molecules. You get transport of these morphogens, and then you get the uptake and degradation of them. And so you get these patterns where you get these gradients, where you get a concentration of morphogen of a certain type, and then it decays over space. And then you get another pattern, you get another concentration here with a decay in the other direction. And then there's a boundary that's formed here, but you can see that, those concentrations as they migrate across, or they diffuse across the cell, they form these stripes. And it's just a matter of overlapping concentrations and the decay of that concentration. And then that sets up a boundary at some point, and you get this striping. So these are ways that you can get self-organization from this kind of control at one scale, which is the molecular scale, getting these patterns at the morphological scale. And then, there are other types of things that they talk about with integral feedback. And this is a very valuable paper, and just in terms of laying out these different issues and kind of going through some of the things in those first two figures in a little bit more detail. They also talk about the management of noise, which is an interesting thing if you're interested in how noise works, because noise is actually very important in a lot of biological systems. Sometimes if you synchronize oscillations or amplify signals, that can be important in determining some of these things, both at the collective scale and even within the cell. So in stem cells and in cells changing their fate, sometimes cellular noise is actually quite important. So there are a lot of different noise mechanisms in cells, and even at the collective scale of phenotypes. So this is all very important. So it really gets into a lot of things that you see in the literature in other areas and brings them to development. So I can send those papers out, but we'll stop here. And let me go to the chat real quick.
Did they mention physical stretch and strain on cells as being part of the morphogens in that paper? I know that there's a paper written about this.
Yeah.
I just wondered, did they mention physical?
I didn't see it too explicitly, but I mean, this is just like examples of this. So they're just kind of hitting examples from the literature, but it would definitely play a role if you think about the different mechanisms. And there are probably mechanisms we don't even know exist to control some of these things, but to draw out those circuits and figure out what are all the factors involved in this. And again, it's different for different systems. So, yeah.
Here I am.
Yeah.
Yeah. I thought I saw a paper about that, but if I run across it, I'll send it to you.
Okay.
The physical being a part of the morphogens. Yeah.
Yeah.
Anyway. Oh well.
Yeah. So
yeah, this is control theory. That whole paper looks like control theory wording to me. So.
Yeah, very much. In systems biology, they do a lot of control theory, but it's usually focused on the molecular scale in systems biology. They're interested in metabolic networks or gene expression networks, and you don't see a lot of papers on physical regulation in systems biology. In developmental biology, you probably will see that more, but that's where they're borrowing that language from. It's kind of coming from control theory. And then systems biology, where it's been modified a bit for biological systems, but not every type of biological system. And then going to,
biological systems are just in between everything and more complicated.
And was that a survey of this field of system biology?
Yeah, well, it was kind of like a survey paper, but it was taking those concepts and rooting them in developmental biology. So it's he goes through a bunch of different concepts from what are essentially things that in systems biology you would see and people read about all the time, but then bring them to a developmental biology context where people maybe aren't thinking about them all the time. And then, it's just kind of that transition. But yeah, it's a nice, it kind of goes over a lot of different, and I'm sure that there are a lot of references in there that are kind of useful for different things. So it would be, I'll send these out later on to the group. We had some things in the chat here. Morgan sent a video. I don't know what that video was, but Alon said, yes, that's the video I've seen yesterday.
Journey to the Microcosmos. I seen it on the Basileria, on Diatoms.
Okay,
And they say some of the stuff that we talked about, that people don't know how they communicate between themselves, with the short range or the long term range communication, how they synchronize their movement, and some of the 1700 guy, a Danish guy that was researching on them. So it's kind of a nice clip.
Yeah. And then, that was it. Then Morgan said, bioink research was the subject of a Terasaki talk last week. Yeah, the Terasaki talks are on YouTube, I think. And they have a lot of really nice stuff on cell biology and organoids and other things like biotech. So, I know Morgan knows about them. But Terasaki talk, if you just Google that, you can find it.
Oh, okay. I should watch some of those just to try to I don't know, bring me up to speed on some of the biology, I guess.
Yeah.
Yeah.
All right. Well, thanks for attending. Yeah. Oh, you wanted to say something? Okay.
I'm good.
All right.
Thank you.
All right. Thank you.
Thank you for the info.
Okay.
All right. Bye.
See you next week.
Bye bye. Have a nice week.
All right.